Okay, we are now at 9:30. I will give everyone just a couple of moments to get back to their desk, seated, ready. And as we do this, I will slowly start to introduce our next speakers. So one will be quite familiar. I'll stop sharing my screen. So we'll be having Bjoern rejoin us, but also I'd like to introduce Austin Crinklaw, one of our research technicians in the lab, to now shortly present T-cell processing and immunogenicity predictions. So I think, Austin, I will hand it over to you. Thank you very much.
Thanks, Nina. So, hey everyone, as Nina just said, I'm Austin. I'm a research tech in Bjoern's lab. Today I'm going to be talking about T-cell processing and immunogenicity predictions. So we'll begin with a little bit of biological background. So here we have a nice little model of a virus. It enters the host. It infects various cells. It can then multiply and spread throughout. And just like every other protein inside the cell, it is subject to degradation. So the viral proteins are degraded into small peptide fragments. These can then bind to class 1 MHC molecules on the surface of the cell. And then so T cells that express CD8, they can recognize this peptide MHC complex, and it can trigger an immune response. So the T cell can either kill the infected cell, release cytokines to recruit other immune cells to the site of infection, and proliferate and generate a T cell memory response. So if we're looking at the kind of set of viral proteins and we're interested in what peptides in this set are actually going to be epitopes and trigger this immune response, we have to understand what's going on inside the cell a little bit further. And and also we would call the peptides that are presented, these would be naturally presented MHC class 1 ligands. So just a little overview of the antigen processing and presentation pathway. So we begin with a protein. It's degraded by the proteasome and cleaved into small peptide fragments. Generally, these are cleaved further into individual amino acids and used for resynthesis. But they can also bind to the TAP transporter and be brought into the ER, cleave further, and then bound to class 1 MHC molecules. These can be brought to the surface of the cell, where they can be scanned by T cells. So ultimately, you have the antigen processing pathway where individual peptides inside a protein are presented on the cell surface to T cells. And so these processes, they're not random. They're specific to certain peptides in the proteins. And the reason that we stress MHC binding is because this is actually the most selective step. Generously, about 2.5% of these peptides will bind to MHC. We estimate that 15% of all possible peptides are generated through the processing pathway. And given peptides that are presented, you can find about half of them in existing T cell repertoires. So I'll begin with the tools that we recommend first, the MHC-NP. So we have two tools available for this for predicting eluded ligands. So MHC-NP, this is actually a tool that was developed by a Canadian group, and this is an example of a tool that is an external tool that we host at the IADB. And also in the same interface, we have NetMHC Pan 4.0. So Bjoern just showed this in the class 1 MHC binding portion of the workshop, but just to kind of reiterate and explain the context for this tool in this case. So since NetMHC Pan is this algorithm that's co-trained on MHC binding and MHC ligand elution data, since it's trained on data from peptides that are eluded and naturally presented, it kind of incorporates the preferences for proteasomal cleavage and TAP transport, as well as the MHC binding affinity. And then moving on to the next tool. So I'm going to kind of work backwards and talk about some more historic tools that we have available. This first one is the combined predictor. It's really a set of predictors that are kind of mimicking what happens inside the cell, the processes that I just talked about. So what you have here is it combines predictions for the proteasomal cleavage, tap transport, as well as MHC binding prediction. And so these predictors, they're trained on specific in vitro data sets, such as proteasomal digest and tap transport efficiency. We can take a closer look at some of the parameters that you can choose. So for the proteasomal cleavage prediction, you can choose your proteasome type. So you have the immuno and the constitutive, where there's some differences in their specificity. And then you also have your parameters for tap transport. So you have two parameters here, and they kind of deal with estimating the length distribution of the precursors available for tap transport. And unless you kind of really know the background and know what these do, we recommend that you leave them as default for best performance. So this is kind of your overall interface. You upload your protein sequences, you specify the alleles you're interested in, and then the parameters I just talked about. Hit submit, and here's your output. So with this output table, you have various scores. You have your proteasome, tap, MHC, processing, and total score. So the higher the score, the higher efficiency for MHC class 1 presentation. MHC binding score is the negative log of the IC50. And then so for the processing and the total score, these are combined scores. They're additive. So the processing would be the proteasome and the tap, and then total score would be proteasome, tap, and MHC. And just as a quick side note, the variances that you see inside this table are going to reflect the differences in the selectivity that I talked about earlier. So for the proteasome, for example, it really, it's kind of evolved to degrade pretty much all possible proteins. So this is not very selective and the variance is low. Meanwhile, MHC is very selective, so the variance you see here will be quite high. And so just to talk about the performance of these predictions and kind of why we recommend that you use NetMHC PAN instead, when we test these scores, the processing predictions, they beat MHC binding predictions, but only when we're talking about predicting eluted peptides. What we don't have is any evidence that the processing predictions are better at when you want to actually predict T-cell epitopes. And one of the explanations for this is that the eluted peptides, they tend to be the most abundant and the most efficiently processed. MHC molecules, their binding preferences, the motifs tend to match what the proteasome likes to cleave and what tap likes to transport. So you have this sort of coevolution. There's a lot of overlap in the top ranked based on binding affinity when you actually look at the processing steps. And another little side note is that proteasomal cleavage and tap transport studies, they tend to have smaller data sets for training. So our recommendation is that you just stick with the MHC binding predictions alone by default. This tool, it can be good if you kind of have limited resources and you'd like to really limit your number of candidate peptides. And then so our next set of tools, it's kind of the same idea, but it has a different approach. It's using neural network-based predictions. And so we have 3 algorithms. The key difference between these algorithms and the previous one is just the involvement in NetChop. And so we have NetChop, NetCTL, and NetCTL PAN. NetCTL just being NetChop, tap transport, and NetMHC, and then NetCTL PAN just being the same except NetMHC PAN. So this is kind of a family of very similar algorithms that build on the family of NetMHC neural networks. And so I'm not actually going to be going through a specific example for this tool, kind of in the essence of time, but the idea of hosting these tools, they're kind of historic predictors. They haven't been updated in quite a while, but we'd like to have them available to those who want to use them. And they can be good if you want to really kind of predict things, or if you're interested in things such as proteasomal digest, but we wouldn't normally use these for epitope predictions. So moving on to the immunogenicity predictions. So what we're interested in here is which peptides are actually likely to trigger a T-cell response when presented. So the idea behind this is that we have this data set of peptides that have similar MHC binding affinity, and you have 2 groups. There's the group that are recognized by T cells and the group that's not recognized by T cells. So we want to find features that separate these peptides. And when we look at the frequencies of amino acids in the peptides that are facing the TCR, in immunogenic peptides, we see an enrichment of these residues that kind of have chemical similarities and characteristics like long side chains, like phenylalanine and tryptophan. And the idea is that these might make these more visible to a T cell. And at the same time, we also see less peptides such as lysine or methionine. So we use this data, these enrichments, to calculate propensity scores. And so here you have your interface, you upload your peptide sequences. Just as a little note, this tool, we only validated it for dimer peptides, but you can make predictions for peptides of any length. And then the parameters that you can choose, the mass position. So these are the MHC anchors. We assume by default that your sequence, the anchors will be the first, second, and the C-terminus amino acids. But if you know that your sequences have different positions for the MHC anchors, then you can upload your own list. And then you submit, and this is your output. You have your peptides, your lengths, and your scores. So the higher the score, the more likely it is to be immunogenic. And the score is just the kind of summation of all the propensity scores at all the unmasked positions. So when we talk about the performance of this, if you look at the scores and you have the two groups, the epitopes and the non-epitopes. So in the non-epitopes, you see this kind of random distribution. And then in the peptides that are epitopes, you do see kind of an enrichment of scores above 0. At the same time, though, you do see some peptides, they're perfectly good epitopes, but for whatever reason, they score below 0. So our recommendation here is that you could use this as a selection step if you want to limit your candidates to the most likely epitopes. And if you're looking for a high specificity, then you should use a cutoff of 0. So just to kind of summarize everything that I talked about, processing predictions, they do work better if you want to predict naturally processed and presented ligands, but we don't have any evidence that they improve predicting T cell epitopes. They can serve as an additional filter and add a little bit more stringency if you have limited resources, for example. And ultimately, we recommend using net MHC pan EL scores, which to our knowledge are the best at predicting binding affinity and is the best approach at directly predicting the likelihood of a peptide to be an epitope. So we suggest you could pair this with immunogenicity scores if you want to kind of further improve predicting epitope candidates. So that's kind of it for class 1. Thank you for listening. I'm going to pass it off to Bjoern and he'll do class 2.
Thanks, Austin. That was cool. I should now be screen sharing and presenting class 2. So let me put on my pointer. Okay. So again, like this is the processing section. So we are in the first section, I talked about peptide binding to MHC, which for class 2 happens here in the lysosomal compartment. And now this is about how do these peptides actually get made that are available for MHC class 2 binding here. So again, like the dominant pathway of peptides that are ending up on MHC class 2 molecules is that they are uptaken from the exogenous space, taken and cleaved by proteases here, and brought into the into this compartment where they can then exchange for clip peptides and bind to MHC class 2 before being transported outside again. So when you go to the T cell tools page at tools.idb.org, T cell tools, you have MHC2NP down here, which is using MHC class 2 ligand elution data to predict natural process MHC2 ligands by scanning the given peptide sequence. So how is that working? As I said, this is based on cleavage motifs that were identified by analyzing what kind of ends do we see? What kind of C and N-terminal residues and motifs around them do we see from peptides that are eluted from cells? So eluted from cells means they are found on an MHC class 2 molecule that is presented on the surface of an antigen-presenting cell. So the assumption is that all those molecules that end up there were cleaved to liberate those peptide from an exogenous protein. So in summary, we have found that ligand predictions can be greatly improved when using binding conditions combined with these cleavage motifs, but we did not find T-cell epitope prediction could be improved with this. And trust me, we tried a lot. So I'll walk you through, this is kind of the summary to make you not too excited about this again. It's been frustrating. We totally would have loved this to work, but we didn't see it. So the tool that we have, it takes a protein sequence, and then essentially what we're predicting at this point, we would have loved to predict T-cell epitopes, but what we're really predicting is naturally processed and presented MHC2 ligands. So it works as normally, you have the input sequence, you submit, and then you get your peptides back. And essentially you see here what it does. So this peptide here has the N-terminal and C-terminal residues. So it's like an S, and then there's the cleavage, and then VP. And then there's P, and then the cleavage, PK, and then the cleavage, and then A. So you can see here essentially the motifs that are found in these proteins, and they get a probability scores corresponding to these residues. Yeah, so it's S, cleavage, VP, and PK, cleavage, A. So that's kind of like the surrounding motif around where the cleavage is being made. So you see that it includes residues outside of the peptide itself. So you're getting a cleavage probability score from this, and then these can be ranked, as we always do against random peptides, and you can get the complete results here that break these down. So when we did this analysis, what we found is we can really get a lot better than just binding predictions by incorporating this cleavage probability scores for MHC class 2 naturally processed ligands. So when we take the binding score and we multiply it by a score alpha, and then we have the cleavage score and we multiply it by a score 1 minus alpha, so then you see this essentially here corresponds to binding score. This is binding score by itself. This is cleavage score by itself, and then this is the combination. And then you see in the combination here, you are getting better than either binding alone or cleavage alone, meaning the specific ligand made can be predicted and is predicted well by analyzing these motifs. So there is essentially certain motifs like having the proline directly preceding. So if the proline would be on the second position here, it will not be cleaved there. These proteases like to have the proline one position apart from the cleavage site. So that is one of the reasons why this has like a high cleavage score. So using this, you really get good at identifying what is going to be the ligand that is going to be eluded from a cell. But what does not work is then trying to use that to predict epitopes better. And this was surprising for a while, and this was our own finding here, but then also Morton's group had a very similar finding, that you could really identify very efficiently the footprint of antigen processing for class 2 natural ligands, but we were unable to use that to make better epitope predictions. And the reason is ultimately that these are not rare motifs, they are found quite frequently. And in a given epitope, for a given MHC binder, you will find a number of possible cleavage sites on the left and on the right of it with very, very, very high likelihood. And then ultimately what's recognized by the T cell and what's bound to the MHC is the binding core. So the binding core is still the dominant reason why a given epitope is recognized. And that's why it dominates knowing the specific ends of the peptide that might be naturally processed and presented. So again, like these scores, we believe work really well of identifying what is really made and presented by MHC class 2. But if you're making a peptide that is longer or shorter and doesn't have the right ends, it'll still be recognized by T cells because the T cells don't actually see stuff that is far away from the binding core. So this is where the tool is. The second part that I wanted to talk about is the immunogenicity predictions. So similar to what we did for class 1, we identified what epitopes are known in the IDB, and we used actually sets of proteins that were tested consistently, like where overlapping peptides were tested for immunogenicity, to have like a really clear yes, no, 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 yes, no, 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 yes kind of pattern overlapping whole proteins, which we found to be more comparable when assigning this training set. And we used this to train a neural network to learn motifs associated with immunogenicity that are independent of binding predictions and independent of the specific MHC alleles expressed. So these were peptides that were overlapping peptides that were then tested in a broad set of people. So the resulting score was intended to be combined with the 7 allele binding method, which quantifies MHC binding across alleles. And then we have like 2 MHC allele independent, essentially, prediction methods that are intended to be used together to identify likely immunogenic peptides. So as I said, we trained a neural network based on our in-house data set, and then we validated it based on tetramers derived from the IDB and 57 independent studies from different groups across the world. This is implemented in 3 approaches: the 7 allele method by itself, the immunogenicity by itself, and a hybrid approach combining the 2. So this is on the IDB page where you get the CD4 T-cell immunogenicity predictions. This should look very familiar at this point, where you enter the sequence and ultimately select your method. These are the 3 methods I mentioned. And you have the similar sorting, selecting, ranking positions, getting an email, etc. And this is the output that you're getting, where you have the immunogenicity score, the combined score, and the median percentile rank. This is the 7 allele method. So all of these are available, and the combined score is essentially using a mixture of the immunogenicity score and the median percentile rank. So, and here you see why, I mean, yes, it worked. So again, you have like an alpha scalar combining the immunogenicity score or the HLA binding score by itself, and then you see here how the combination of the 2 works better. But you're looking at AUC values of 0.725 versus 0.70, which is, I mean, it might sound, before I was making a big deal of 0.96 versus 0.97 as being a big difference, but that's because you're close to perfection, getting much better, much closer to perfection in terms of like impurity, versus here, you are far away from perfection, get a little better. So we think this works, and in principle, there are some motifs there that are associated with immunogenicity, but for things like that, we typically apply Occam's razor, and that says we don't actually think that using these combined predictions are recommended, given that the improvement is really minor, and we like to keep things simple if we can. And for epitopes, if you want to do predict epitope candidates. So we think this works, and does give some improvements, but it's just not very much. And we are always worried that we are introducing biases that we are not aware of. So we don't necessarily recommend these and would stick to just the MHC binding predictions by themselves. If you do want to predict naturally eluded ligands by themselves, absolutely use these. These give you significant improvements that are very clear and clearly motivated. So ultimately, let me stop here. Both class 1 and class 2 epitope predictions are constantly being reevaluated. As I said before, that includes the processing predictions and these recommendations, as everything else we're giving you, are potentially subject to change. And again, like, let me just say also for class 1, this was my PhD. I worked on trying to get processing predictions incorporated, and I'm showing how they get better at predicting epitopes. And I had to admit myself that this is not the best approach. So I would like to see these work if anyone does. And I still think this is a really amazing work of research, but ultimately just MHC binding by itself is a solid, useful tool that gives us pretty much what we want without any kind of potential sources of bias and complex rules. And I'm always preferring simple and robust over complicated and fancy. And that is it. I think we're a little early.
Yes,
Yeah.
thank you, Bjoern. Yeah, just a couple of minutes early, but no problems there.